Anyway, I'll start sharing the screen and then I'll start the presentation. So maybe the others can see it on the recording. Is the screen visible in full screen mode?
Yes, it's clear.
Okay, yeah, thank you. So this is the 11th session. We just have one more session which will happen tomorrow. Like I mentioned, I'll not be available in the next week since I'm traveling. So I'm finishing the 11th and the 12th session on consecutive days, today and tomorrow. So here the date still says 12 because it was actually planned for tomorrow. But today and tomorrow I'll be finishing this session. So yeah, today this is going to be a continuation of what we have previously looked at, how metal contamination can be cleared up using biological organisms, microbes specifically. Let me just confirm that the recording is happening. Yeah. Biohydrometallurgy. This is nothing but another term for biomining. Biomining is a process of obtaining metal from ores or mine waste using microorganisms. So usually people go and mine metal ores or natural places where these metal ions are found, and then they get these ores. And after the ores, through series of chemical processes, the metal is extracted. So biomining is also a similar process where instead of using chemical processes, here we use biological organisms which can precipitate metal ions, and we can get metals from them. This could also be mining waste because after a certain point, it can't be directly mined out. So in such cases, there is a lot of mine waste accumulation that happens. That can be used, and the metals can be extracted from such mine waste also using microorganisms. And this doesn't have to be just something like a mine waste. Just a second. Yeah, sorry. um, yes, the screen still visible in full screen, right?
Yes, clearly.
Yeah, okay. So this can also be used for places which are contaminated with metals, like wastewater, or any of the soil that is contaminated with metals can also be used as the starting material, and microorganisms can be applied on them, and the metals can be extracted out and purified from there. So this is also increasingly being used as an application of biotechnological processes, where it can play a role in the mining industry. Biomining technique, like I said, is used to clean up sites polluted by metals. So this is an example of a copper mining site. So after they mine for copper, there are still residues left out in this region in the mining site, and that region is not really suitable for any other purposes because that region is still contaminated with copper metals. And we have seen in one of the previous sessions that copper accelerates some of the catalysis process for microorganisms, but it becomes deadly or lethal after a certain higher concentration. So therefore it is important to understand what exact microbes can be used in a particular mining site or a particular contaminated site, so that it can effectively precipitate the metal that is present. So this is an example of how the process of biomining happens. Bio-leaching is a process which utilizes metal sulfides. So this is a sub-process of biohydrometallurgy or biomining, which is bio-leaching, where specifically metal sulfides, so metals that are in the form of sulfides, are used as a source of energy, and ferric compounds are used as a source of electron donors, and various bacteria can convert the Fe2 plus to Fe3 plus, and then reduce them and so on. And the copper sulfide, so if it is a copper based mining site, and if there are copper sulfide ores, bacteria can be used to extract copper, and in the presence of Fe2 plus, and converting it into Fe3 plus. So these are again reduction and hydrolysis reactions that happen in this process, and this compound is taken up by the bacteria and processed to obtain Cu2 plus. So what is the difference between biomining and bioleaching? So this is like a sub process. So biomining is a technique where we can extract metals with the help of prokaryotes and fungi. So prokaryotes are a broader term for various organisms, but bioleaching is specifically for bacteria, and it can't be done by fungi. So this bioleaching process specifically applied to certain bacteria that can act on metal sulfides. So here, like I said, prokaryotes, any prokaryotes or fungi can be used. So here specifically iron sulfur bacteria are used because this bioleaching process involves conversion of metal sulfides and using iron as a electron donor. So specifically bacteria that act on iron and sulfur will only be useful in this process. Microorganisms can take up metals in the ore into cells, and we can extract the metals from the microorganism cells. Whereas in bioleaching, bacteria are further used for oxidation of the metal ore, which isolates the metal and allows to extract the metal from the ore. So the major difference between biomining and bioleaching is that in process of biomining, the microorganism takes the metal compound into its cell, and the processing happens inside the cell of the microorganism, and whatever gets precipitated is the metal of interest. However, in case of bioleaching, it is mainly an oxidation process. The oxidation process happens outside the microorganism cell, but it has enzymes that can oxidize the metal in the ore, and after oxidation, the metal can be extracted out from the ore. There are different ways this bioleaching can be done. These are the major three ways through which bioleaching usually happens. The first thing is called slope bioleaching. So here this is usually done at a larger scale, like the image that I showed for a copper mine, which is like really big in size. They spread the ore in a slope like fashion along a mountain or along a vast area, and the indigenous microorganism in that region is stimulated so that it can convert the metal, so it can oxidize the metal along the slope, and then it is extracted. However, in the second case is the heap bioleaching. Here, this is also done on a larger scale, where the the ore from which the metal has to be extracted is layered in the form of a heap, like multiple heaps of the metal, or the metal ore is laid on one top of the other, and the metal is recovered from that using the microorganism by adding the microorganism and enriching it with other nutrients that it would require, or the energy source and electron source for the microorganism to act on the metal. The other process is the in situ bioleaching. This is a process where instead of artificially layering it like a slope or as a heap, the ore is left in the place where it is, but the microorganism is added inside the place where the metal ore is present in a very systematic way. Like you can see, like this kind of step like fashion it is created, and the microorganism is added inside this. So whatever the extraction happens will happen inside too. Inside too means in that given site itself, where the whole ore is present, and the extract after the reduction or the oxidation process happens, the converted metal is extracted out from the site. So these are the three major types of bioleaching process. So from here we go to another process called EBPR. So we will see what EBPR is. But before that, we will give a quick recap for what eutrophication was. We saw this in a couple of lectures before. Eutrophication is a process where, because of higher amounts of nitrogen and phosphorus being enriched in water bodies, the algae starts increasing in numbers. This is called as algal bloom. They start consuming all the oxygen that is dissolved in the water bodies, and they start increasing in number. Phytoplanktons also take up this oxygen. They also use this nitrogen and phosphorus and start growing. So these algal bloom and plankton growth will now reduce the amount of oxygen that is dissolved in the water, and because they're growing on the surface of the water, they also take up all the sunlight that is coming into the water, and they prevent the sunlight from reaching inside the water to other organisms that require sunlight. So this reduction in dissolved oxygen actually is very detrimental to fish and other organisms that live in water, other plants that grow, that use water as a substrate for growing, so on and so forth. So eutrophication is a very lethal ecological problem, where it completely destroys the water ecosystem, and specifically promotes algal bloom in water places. This is because of the increased amount of nitrogen and phosphorus that gets mixed in this water, mainly because of agricultural runoff, or it could be because of nitrogen and phosphorus from industrial waste that gets mixed with the water, and this causes loss of food habitat and oxygen production in the water bodies. So why do we have to now come to eutrophication? How does it have any application here? Because microbes can again help in controlling this process, can avoid this harmful algal blooms from happening by removing the excess of nutrients such as nitrogen or phosphorus that gets mixed with the water. Certain bacteria that can use this as nutrients for their growth and process them into something that can be easily fixed, is what we are trying to focus on in this. So this graph just shows how eutrophication can be harmful, how it can lead to harmful toxin product formation from certain organisms, which can destroy fishes, birds, and mammals. It can result in hypoxia. It can This is because of nutrient loading and organic loading is what is the trigger for all of this. This can result in foam and slime formation because of this excess nutrients. When they mix with water, they can start forming foam. This is specifically in case of soap like substances which cause detergent like substances, which are usually washed off from industries, cause this foam or slime formation. This will now again prevent sunlight from reaching the bottom. Oxygen will get locked between this, and it will keep foaming more, and it will not pass through. The water temperature also changes, oxygen gets depleted, so on and so forth. So these are some of the harmful effects of eutrophication, which can be fixed by microorganisms. So EBPR, which was the beginning of this slide of this particular chapter, the expansion is enhanced biological phosphorus recovery. So nitrogen and phosphorus, or like I mentioned, are the major problems in these wastewaters that gets contaminated during the wash off. So during the biological phosphate removal process from wastewater, phosphates gets accumulated in microbial cells as polyphosphate granules. So we specifically choose microbial cells that can take up this phosphate and convert it into polyphosphate and make this as granules and store it, or pass it on, or it can be processed later on by some other microbial organism. So this is done as a three-step process, where initially it's an anaerobic process followed by anoxic and aerobic condition. So here there is no presence of oxygen, and in this condition the microorganism takes up phosphate, and here a phosphate rich substrate gets precipitated, and the sludge can be removed and sent back as an effluent without this higher phosphate content. So that forms the major crux of the enhanced biological phosphorus recovery process. So usually this process is tried is done in many of the sewage treatment plants, where there's a lot of phosphorus and nitrogen content from human waste and other municipal waste and so on. In such conditions, instead of just treating the sludge and removing the bio waste and the biomass, yeah, it would be better to enhance this with microorganisms that can specifically remove phosphorus and nitrates. So in this process, we require two different types of microorganisms, not just phosphate accumulating organism. There should also be a glycogen accumulating organism, because whatever the complex products are there, the volatile fatty acids, everything has to be broken down, and the phosphorus has to be sequester, and in the meanwhile glycogen will also be produced from the phosphorus accumulating organism, which has to be broken down by a fermentative organism such as a glycogen accumulating organism. Polyhydroxyl alkanoates, PHA, are produced during the anaerobic condition for the enhanced phosphorus removal. So here, before it passes through this, there is an anaerobic process, and during this process, PHA so produced and glycogen is produced, and hence this glycogen has to be converted by another microorganism before being removed for the effluent discharge. So here, these are different types of bacteria that can take up this role of the fermentative accumulation, fermentative glycogen accumulation, or the classic accumulation of phosphate and glycogen. And the next process is biological nitrogen removal process. So here in this process, there is again a necessity for two different microorganisms. So all of these processes, as we've been seeing from the beginning, let it be the sludge removal before the waste treatment treatment, where the phosphorus has to be removed, or even the nitrogen has to be removed, two different bacteria have to work in tandem. So here as well, a nitrification bacteria and a denitrification bacteria have to work in tandem. If either of one of the organisms is not there, and if we are enriching certain bacteria and depleting another bacteria by using substrate that are not correct, then at the end of the day we will not have the final end product that is desired. So here in the case of nitrogen removal, there are nitrates that are released or discharged into the water bodies in the form of fertilizers or human waste. So these nitrates need to be fixed and converted to nitrogen gas, which can be released to the environment. So that is the end goal, because nitrogen gas is the most abundant gas found in the atmosphere, and hence that's not harmful to the environment, and hence it can be released. So here in this process, first the ammonia, whatever that is in the form of a fertilizer, the ammonia has to be oxidized by nitrifying bacteria and converted to nitrite and then to nitrates, and then the nitrates have to be converted to nitrogen gas by denitrifying bacteria. So this nitrification and denitrification is also a process where we have seen, even in the case of nitrogen fixing bacteria, which help in fixing nitrogen for plants. So these work in the opposite direction. They take nitrates and then or ammonia in the soil, and then they fix it for the plants so that the plants can take them in a way that it's easy for them to take in. So here in the case, we have to work on the other way. Whatever ammonia is there, it should be broken down into nitrogen gas for release into the environment. So here in this case, enriching the ammonia oxidizing bacteria and denitrifying bacteria is important, because ammonia is getting converted to nitrate and then to nitrite and then getting converted to N2 gas. Here if we do something in the opposite way, like we enrich bacteria that instead of oxidizing ammonia, that reduces ammonia, or if we use a bacteria which instead of converting nitrites back into N2 gas, it is fixing more nitrogen to nitrates. In that case, if we use any mismatch of bacteria, then the process won't be achieved as the way we want. So biological nitrogen removal process should happen in this sequence with these particular bacteria. So this is the process how the nitrification happens. So here the bacteria which can perform this oxidation of ammonia is called as anammox. That's the bacteria is anything that can fix nitrogen and can convert ammonia to get it through the process of assimilating ammonia, to get it through the process of denitrification and convert to nitrogen, is called as an anammox bacteria, and that is the most important component of this whole biological nitrogen removal process. So this is how the conversion of nitrogen, the conversion of ammonia to nitrogen happens inside the cell of the microorganism. So here, this region where the ammonia conversion happens is called as anamoxosome. Anamoxosome is an organelle where ammonia is processed by anamox bacteria. So this is again very similar to electron transport chain reactions that happen inside any biological organism for converting an energy source into electrons and then ATP for energy. So here it is happening in the riboplasm, where the microorganism has different enzymes. These three enzymes specifically, which help in the conversion of the ammonia that is being taken up by the microorganism. So yeah, so these are actually what is there in the session 11, actually the 11th week. These are all is what mostly covered, biohydrometallurgy and how the phosphate can be removed and nitrogen can be removed, and how these different bacteria have various enzymes and specific regions that are required for conversion of the nitrates into nitrogen and help in fixation. So that's pretty much what I have for today. So if there are any questions, um, so please do ask if there are any questions.
One question that we are giving that enhanced phosphorus, biological phosphorus removal, right?
Yes,
There we can add anaerobic bacteria or facultative anaerobic, because mostly it will happen in the water region, right?
yes.
Reflex removal. So the water will also contain the dissolved oxygen anyhow, minimum amount also.
Yes.
So the faculty anaerobe will be the better choice, right?
Yes.
Like
Yes. Yeah. Yeah.
process. Okay.
But these are also cases where the extreme cases where we already assume that the oxygen is being depleted by algal blooms and so on. So the oxygen content available itself is very minimal. But yeah, the best choice would be to go for facultative anaerobes, which can still have some oxygen in that and still perform the reaction. Yeah.
Okay. Hope
Any
you
other
have.
questions?
No, thank you. Hope you have.
Okay. Thank you. So uh, see you tomorrow around the same time, 6 to 7.
Okay.
Tomorrow also will be a shorter session only because the last few weeks of the content is very less. It's not uh,
Yeah.
like many slides. So if, I mostly want if there are questions from other weeks or other sessions, if there are any questions, so please do ask because tomorrow will be the last session for the problem solving session, and then there will be the exam.
Okay. Sure. Sure. Thank you.
Okay, thank you so much, sir.
Thank you.